Let's move on to talking about the tissue systems. There are three main tissue systems in the plant body. We'll go over each of these in more detail. The dermal tissue, dermal, the same word origin as human epidermis, which is the skin or the outside layer. The vascular tissue, which include conducting cells and also supporting structures. And the ground tissue, which is kind of thought of as the meat of the plant and is often defined by location. The dermal tissue consists of flattened epidermal cells plus some additional specialized cells. The dermal are, again, on the outside layer of the plant body. In the roots, the epidermis consists of root hairs, which are the specialized cells. In the root, these root hairs are individual cells that have become extremely elongated, as you can see in this little diagram of the root ceiling to the right. These root hairs help to increase surface area for absorption of both water and different minerals. So having lots and lots of these root hairs that are constantly kind of being new ones arising, all these root hairs increase the surface area similar to how villi and microvilli do in animal intestines. Increasing surface area, allowing for more absorption. The epidermis in the leaves consists of a cuticle on the outside, and that's an outer waxy layer that helps to prevent water loss, especially. There are also stomata or stomates embedded within the leaf epidermis, and these are those leaf pores that the plant actively opens or closes to allow water loss through the leaf, and also the input or influx of different gases. The epidermis also contains fibers and trichomes. These trichomes are these leaf hairs, also these single-celled projections, and they can have some different or cool shapes. Like this one over here is a three-parted gland trichome, and these are involved in defense against herbivores especially. Some of these can also be glandular trichomes, which emit or produce different sticky substances. The leaf epidermis can also contain different pigments and defense compounds to protect the leaf from herbivores and also from excess sun damage. The vascular tissues are xylem and phloem, and these develop from the procambium, which is that primary meristem. For the development of xylem and phloem, if you think of kind of the procambium as a line like this, it produces xylem to the inside of the plant and phloem to the outside. So if we have the inside of the plant here and the outside here, produces xylem to the inside and phloem to the outside constantly. So constantly simultaneously dividing in both directions. In dicots, these are often found arranged in vascular bundles. So remember what the difference between monocot and dicot. One of those was how the vascular bundles were arranged in the stem. In monocots, they were scattered, and in dicots, they were found in a ring around the outside. So each of these then is a vascular bundle with xylem on the inside, phloem on the outside, and the vascular cambium in the middle. We will talk about these a lot more when we talk about nutrition and transport. But briefly, xylem tissues conduct water and minerals. Xylem is made up of several different cell types, not just one cell type, including both sclerenchyma and parenchyma. So different cell types can make up one tissue type. Xylem usually have thickened cell walls. You can see, especially down here, these xylem cells have really thick cell walls, and xylem are used especially in water transport and support. This lignin in the cell walls really helps that support as well. Here we have xylem. We have two different types of xylem, vessel elements and tracheids found in gymnosperms or angiosperms. Xylem are also dead at maturity. So these are cells that tend to be dead at maturity, and they're the most abundant cell in trees, which we'll discuss in just a second. Phloem, so phloem down here, is involved in the transport of organic molecules and also some minerals. There are also several different cell types found in phloem, both sclerenchyma and parenchyma, and different specifics called sieve elements and companion cells. Again, we'll discuss those in more detail later. the phloem are usually alive at maturity as well. So these vascular tissues include xylem and phloem. They're derived from the procambium or vascular cambium, producing both xylem to the inside and phloem to the outside. Ground tissues are the third tissue system. These are derived from the ground meristem and are defined by location. So they can be located between the dermal and the vascular tissues. So here we have D for dermal, V for vascular, and G for ground in this leaf example. In the stem example, they'll often be found in the center. So here we have ground tissue both here and here, with vascular bundles found here and dermal again on the outside. And in the root, the vasculature is often found on the inside, and the ground tissue is then again sandwiched between the dermal layers and the vascular layers. These dermal tissues are involved in supports. They're also the primary site of photosynthesis in leaves. So again, remember the mesophyll containing those two different types of parenchyma, those are ground tissues. So we have cell types, which are parenchyma, found in the tissue, which is ground tissue here. They can also be used as storage tissues. The cortex of the stem also largely consists of these parenchyma cells. This is storage area for different starches. And since these cells can still divide, they're often involved in wound healing as well. The pith, so that's a little messy. The pith also originates from the ground meristem. It's composed of spongy parenchyma tissue that's additionally modified for storage, but has very little structural strength. So this is usually located in the center of the stem of dicots. It's encircled by a ring of xylem or woody tissue or conducting tissue, and outside of that, a ring of phloem in some woody groups. That leads us to the idea of secondary growth. Primary growth comes from the primary meristems. Again, those are the procambium, the ground meristem, and the protoderm, whereas secondary growth comes from lateral meristems. Primary growth adds length, secondary growth adds girth. In herbaceous plants like beans or corn or a lot of annuals, these produce primarily the primary vasculature, whereas woody plants produce both primary and secondary vascular tissue. If you recall, there's a giant tree trunk sitting in the 223 classroom, and you can really see what the secondary growth looks like. We can think about the rings of the trees and all of this woody growth, through here. This is all secondary xylem. For instance, and what this really does is this secondary xylem gives a lot of support to different woody growth, especially. So in 2006, this redwood named Hyperion was discovered in Redwood National Park, and it's considered to be the tallest tree and really tallest living thing in the world, measuring about 379 feet. So with the evolution of secondary growth, vascular plants could develop these thick, strong trunks that allowed for greater heights. There's a video embedded here if you would like some more information on Hyperion and this attempt to climb the world's tallest tree. Lateral meristems increase girth. That's produced by these woody plants. So woody plants start with herbaceous growth from primary meristems and then go on to have the secondary growth. So if you look at this kind of cross section of this woody plant material here, we have remains of primary xylem and pith on the inside. If going from inside to outside, so remains of the primary xylem and then pith, and then secondary xylem, the vascular cambium, and then phloem here. So the vascular cambium again produces xylem to the inside. To the inside, you got X for xylem, and to the outside, you have P for phloem. So xylem to the inside and phloem to the outside. So as you continue moving, you get the phloem, and then because we're expanding outwards continually, so if you think about continual expansion outwards, what used to be there then gets crushed. So the current year's secondary phloem will continue to expand outwards and actually crush the older primary primary and secondary phloem. Outside of this, we have cork cambium, which is that lateral meristem derived from the ground meristem, and outside that cork, which is also called the outer bark. If you take a different look at this, for primary growth, again, you have an epidermis on the outside. You have a vascular cambium through here, producing xylem to the inside and phloem to the outside. As that continues into secondary growth, you now have lateral meristems with phloem to the outside, xylem to the inside, and so you can see we continue to get pushing out and out. That continues to go out. You get more and more layers of secondary xylem, and all the secondary xylem is the woody tissue, and then the primary phloem is still pushing out against the primary xylem. All of these secondary xylem layers that are produced every year produce what we call annual growth layers. Secondary xylem layers form these growth rings that we see in trees. You've probably seen examples of this and how you can age trees by looking at their growth rings. This is only secondary xylem, not from the phloem. And the reason that you can really see these rings, and it's not just kind of a continuous thing, is because of differential cell expansion in the spring or early in the growing season compared to later in the growing season. So in the spring, growth is usually pretty fast and there's lots of cell expansion. And this results in the spring xylem having larger cells. And the vascular cambium here is producing xylem to the inside. During the summertime, we still have that vascular cambium found here. In the summertime, growth slows down and there's less cell expansion, and so you get summer xylem that are smaller cells again. And then the next year you get another layer of spring xylem, and that results in kind of this differential pattern of, here we have one ring with your summer or your spring xylem and your summer xylem, and then a new year spring xylem, and this continues on and on. Again, this is all that secondary xylem forming these growth rings. Additionally, so what that looks like is you got this cork layer formed here with the outer bark here. And that cork, you might know, is really kind of a cushiony substance and is used for popping your bottles. So again, the other lateral meristem that we have is called the cork cambium. So the vascular cambium and cork cambium are two lateral meristems that help to increase girth. In the cork cambium, we have, so here's the cork cambium. The cork cambium produces cork. And this corky layer is located right here. It surrounds the secondary phloem. So here would be the secondary phloem down here. The cork cambium produces cork, which replaces the outer epidermis with outer bark as kind of the epidermis ruptures, increasing girth. So again, we're constantly increasing girth, pushing from the inside towards the outside, which eventually will burst through the epidermis, and we got to replace it with something. These cork cells are dead at maturity, and they're also layered with lignin and suberin. So we have lignin and suberin, which are these hard layers, and what they do is they provide strength and also water tightness, not allowing things to get in. This helps to defend the plant against microbial pathogens as well. So in the actual plant, you can see here we have layers of secondary xylem. The vascular cambium would be in here with the phloem, and here we have the cork and then outer bark layers. This cork is often kind of squishy material. You can feel some of those other compounds in there, that lignin and that suberin. And that cork, of course, is used for pop-in bottles.